Ah, Vamos a dar comienzo una vez más a este ciclo de conferencias de la Academia Nacional de Agronomía y Veterinaria. Para abrir el ciclo de conferencias este año, del año 2026, tenemos realmente una invitada de lujo, una verdadera experta en el tema que va a ser presentada con todos los detalles por el académico Alejandro Schrudel. Simplemente decirles que Linda, como ustedes ven en la transparencia que se está proyectando, es un investigador, es una profesora de la universidad y hoy va a estar presentándonos lo que es la transmisión zoonótica e interespecífica de los virus de corona o coronavirus. Así que, dicho esto y con muy pocas palabras y para no perder tiempo, le doy la palabra a Alejandro para que nos haga una presentación de quién es la persona a la que nosotros vamos a escuchar en los próximos 45 minutos, tras los cuales abriremos un espacio para que le hagan llegar a ella las preguntas. Si alguno quiere preguntar en español, nosotros podemos traducirlas y si no, directamente pueden preguntarle a ella en inglés. Me piden por favor la palabra este, en el chat y yo voy otorgando en orden esa palabra. Dicho las reglas del juego, vamos entonces, Alejandro, un gusto que nos presentes a la doctora Lindra Saif.
Gracias, ingeniero Satorre, y buenas tardes a todos. Bueno, yo le pedí a Linda un corto resumen de su actividad que me lo pasó en inglés. Voy a tratar de traducirlo, pero aún, además de traducirlo, abreviarlo, porque la trayectoria de más de 40 años de la doctora Saif realmente no se pueden compendiar en 2 minutos, pero lo voy a tratar de hacer en los aspectos más relevantes. La doctora Saif es actualmente profesora honoraria y distinguida de la Universidad de Ohio State University en Estados Unidos, en el Centro para Estudios de Alimentos Animales en el Colegio de Alimentación, Agricultura y Ciencias Ambientales y Veterinaria y Medicina Veterinaria. Es además codirectora del programa que tiene la universidad sobre patógenos emergentes, particularmente los virus. Ella obtuvo su bachelor y su máster y su PhD en microbiología, todo en la Universidad de Ohio, y está radicada en el Centro para la Alimentación de Worcester, que es un centro que tiene una trayectoria enorme de los últimos 50 años en virología y en inmunología relacionado con virus del aparato respiratorio y del aparato digestivo. La actividad de Saif es tan relevante que es un laboratorio reconocido internacionalmente por organismos como la WHO y el CDC y referente internacional en temas relacionados con estos virus y con la respuesta inmune a estos virus. Así que yo creo que hablar de Linda Saif, de la doctora Linda Saif, es realmente hablar de una eminencia internacional. Es miembro de la Academia Nacional de Ciencias de Estados Unidos, de la Academia de Inventores de Estados Unidos, de la Academia Nacional de Microbiología, y tenemos el honor de tenerla también como académica correspondiente en Estados Unidos. Yo voy a tratar, Linda, de resumir los dos aspectos fundamentales que hacen a su trabajo. El primero es una investigadora de primer nivel, más de 485 trabajos en las mejores revistas referenciadas internacionales en virología y inmunología. Ha escrito más de 85 capítulos en libros relevantes, relevantes que son libros de la biblioteca de un laboratorio especializado en cualquier parte del mundo. Así que desde el punto de vista de la investigación es relevante en todo sentido. Y siempre trabajando con estos virus, los corona, los rotas, que la llevaron a ser una referente internacional cuando apareció el SARS-1 en el 2002 y luego el SARS-3 con el COVID y el MERS, que es un virus parecido también, pero que ocurre en Medio Oriente. Y en todos estos temas ha sido su aporte y el de su laboratorio y sus colaboradores ha sido relevante. El segundo aspecto que yo reconozco en la doctora Saif como muy relevante es la formación de recursos humanos. Ha formado líderes de opinión en investigación y que son líderes en laboratorio de investigación en Europa, en Asia, en África, en América. Y a través de esa actividad es que nos relacionamos hace más de 40 años en la Argentina. Hay académicos nuestros que tomaron sus maestrías y su PhD en su laboratorio, trabajando en conjunto durante mucho tiempo. Así que hablar de la doctora Soy con nosotros es hablar de una eminencia científica relevante. Yo quisiera terminar nada más que es una investigadora que se ha sabido adaptar a los tiempos y la presentación que nos va a dar hoy es una presentación que hace a lo último en estos virus que nos están afectando, pero tiene una proyección no solamente para estos virus, sino para el rol de los virus en la transmisión interespecífica que hace a la evolución de la naturaleza. Así que yo espero con ansiedad la presentación de Linda. Linda, I have made some arrangements on your CV, but this is my responsibility. So I leave you with your presentation, and we expect it, like always, an excellent presentation. Thank you very much for coming, for attending.
Thank you very much for the introduction. It's wonderful to see my colleagues and friends from Argentina again, even if it's virtually this time. And so I'm very honored to be able to speak to the Academy and the Academy members. So with that, I'd like to go ahead and present my slides. So the title of my talk is Emerging Coronavirus, Their Zoonotic and Interspecies Transmission. So coronaviruses, for those who are not very familiar with them, although I think everyone is after the SARS coronavirus 2 outbreak, the COVID-19, the coronaviridae family has 4 genera called alpha, beta, gamma, delta. They're positive sense envelope single-stranded RNA virus, the largest RNA viral genome, 30 kilobase. And the spike shown here schematically and on the surface of the particles in the electron micrograph encodes the virulence and receptor binding domain epitopes, and it's the vaccine target for neutralizing antibodies. So all of the RNA vaccines that were made to COVID-19 were directed toward the receptor binding domain in this spike. It infects mammals and birds. It causes mainly respiratory and enteric disease, but it can cause other syndromes that are noted here, including some systemic syndromes. The viruses all have similar genomic organization with a large replicase at the one end, and then at least four structural proteins have been identified that's encoded in each of these single genes, the spike, nucleocapsid, envelope, and membrane. And some coronavirus also have a hemagglutinin shown here for HKU1 strain. The difference among these viruses, even though they have a similar genomic organization, they all have different numbers of these accessory genes shown here in the purple color. And the function of many of these are unknown, but they're thought to encode genes, proteins that can shut down the host innate immunity. So these are the questions I'd like to address related to the SARS coronavirus 2 and animal coronaviruses. Why are coronaviruses so diverse? How do they emerge and evolve to infect new hosts? Also, how do they accomplish interspecies transmission? What are the potential animal reservoirs or hosts for SARS coronavirus 2? How is SARS coronavirus 2 transmitted from animals to humans as a zoonosis and vice versa as a reverse zoonosis? And so I want to talk a lot about the reverse zoonosis as well. That's not as widely understood. Another question is, will coronavirus 2 persist in a multi-host reservoir? Human to animal spillover, and then animals spill back to humans, even though we think of SARS coronavirus 2 more as an endemic infection now. And last, I want to review something about the role of One Health, which WHO defines as an integrated, unifying approach that aims to balance and optimize the health of people, animals, and ecosystems. And this plays an important role then in preventing and mitigating pandemics. So the first question, why are coronaviruses so diverse? How do they emerge and evolve and persist to infect new hosts? Well, one scenario is they have this very large RNA genome. So they have high plasticity to accommodate mutations, insertions, deletions, any changes. They occur as a quasi-species or a swarm of viruses in a population. And this could explain the origin of these interspecies variants with altered tissue or cell tropism. And I'll mention a couple of these in my talk. The enteric coronavirus TG in swine and mutated by a large deletion in the spike to a respiratory variant, the PRCV. And we know that feline enteric coronavirus can mutate within a cat to highly systemic strains that infect macrophages and cause a fatal disease, feline infectious peritonitis. And we also see somewhat different tissue cell tropism with the Omicron sublineages, more upper respiratory tract versus the other variants that are upper and lower respiratory infections. Mutations in the spike and recurrent deletions can alter antibody epitopes, but when you have a deletion, it can't be corrected by the coronavirus proofreading activity. And we also see in coronavirus very high recombination frequency, as high as 25% in mixed infections. So mutations, deletions, and recombination can lead to the emergence of new strains. And I want to provide some examples of these during my talk. So this is a phylogenetic tree of human and animal coronavirus based on complete genome sequences. The blue ones are alpha coronavirus. The gold ones are beta coronavirus. The peach colored ones are gamma coronavirus, and the beige ones are delta coronavirus. So the ones that I want to talk about today are in the alpha coronavirus genus, the pedicovirus or porcine epidemic diarrhea coronavirus that I'll mention, the rhinovirus, the severe acute diarrhea syndrome coronavirus in pigs in China, and then several of these tegacovirus, including the recent human pneumonia strains that spilled over into humans from dogs. And I'll also talk about beta coronavirus, the embecoviruses, particularly the human strains here that have spilled over from cattle or into humans. These two strains are rodents into humans. And then the SARS coronavirus 2. I'll spend most of my talk on time talking about SARS coronavirus 2, but also the porcine delta coronavirus that is also now shows examples of two spillovers, likely directly from swine into humans. So this is showing the projected origin and timeline of the emergence of the human coronavirus. So for the alpha and the beta coronavirus, the Sarbeco and Merbeco ones, the reservoir host is thought to be bats. But for some of these other beta coronaviruses, the Mbeco viruses, it's thought to be rodents through intermediate ungulate hosts, particularly cattle for OC43. And then for porcine delta coronavirus, it's thought to be avian reservoir host. So if we look at this timeline down below, we see these alpha coronavirus. These are the common cold coronavirus that have been with us for centuries, that originally the NL63, it's thought to have spilled over directly from bats to humans centuries ago, and 229E from bats through camelids, including alpaca strains to humans, and all of these now transmit human to human. And then for the beta coronavirus, we have these historic strains of common cold coronavirus HKU1 that arose again centuries ago, likely from rodents. We don't know if there was an intermediate host. And then the more recent of all of these common cold coronavirus, the OC43, that arose in 1898 approximately. This is all molecular clock analysis, spillover from cattle to humans, and human-to-human transmission. It's interesting that this virus, the time frame that this virus dates back to was during what's called the Russian flu pandemic. And people thought this was influenza, but it was never proven to be influenza. And many people now think that this actually could have been, because of the time frame, the first coronavirus pandemic. So we have here, for the past 2 decades, we've seen the rapid spillover of severe coronavirus respiratory diseases. The first one, SARS coronavirus, 2002 to 3, from bats to civet cats in wet markets to humans with human-to-human transmission. Then we had the MERS coronavirus in 2012, from bats through camels to people with human-to-human transmission that's ongoing in the Middle East, occasionally escapes, and the most recent cases were in France. And then we have bats. Potentially, we don't know the intermediate host for SARS coronavirus 2, but again, potentially raccoon dogs or pangolins in wet markets. So we have emerging coronavirus strains that have recently spilled over to humans too, that I'm going to mention. These could represent WHO disease X. These include the human pneumonia, human porcine delta coronavirus associated with systemic mild infections in children in Haiti, with 3 strains identified, likely from pigs directly to humans. And then we have the human canine coronavirus, likely from dogs, spillover into humans that were discovered in 2021. So we now know from studies of animal coronaviruses, and if we take swine coronavirus as an example, and I will use this as an example because I've spent my whole career studying swine coronavirus dating back to TG, although not dating back to 1946. But all these pink are the swine viruses I'm going to mention. I think it's important to study these and to understand how mutations, insertions, deletions, and recombination have generated these porcine coronavirus over time, with altered virulence, tissue tropism, and even potential for cross-species infections. So I want to mention some of the lessons learned from studying these porcine coronavirus and the potential scenarios for SARS coronavirus 2 evolution. So first, when studying TGE and PRCV, TGE is an enteric virus, originally spilled over from dogs to pigs. And it was stable, very stable for at least the past 40 years period. And then a variant of this virus arose spontaneously called porcine respiratory coronavirus. It had a large deletion in the spike, and it changed the tropism from enteric to respiratory and spread all through the United States, Europe, and then Asia by the respiratory route. And this is similar to what we see with the Omicron variant of SARS coronavirus 2, where it's acquired more of an upper respiratory tract tropism now. We also know that the viruses can emerge from bats for PEDV and the SARS coronavirus in China to cause either a global epidemic like PEDV. This virus first appeared in Europe and became widespread in Europe and then largely disappeared, but then it reemerged in Asia, and then it spread to the US in 2013, where it killed approximately 8 million baby pigs. And we know that these established viruses, like PEDV, can disappear and reemerge in other regions, as I just mentioned, with increased virulence and mortality in pigs. And we see this situation where we had SARS. SARS largely disappeared, although it's still present in the bat reservoirs. And then this virus emerged as a sister to SARS, as SARS coronavirus 2, about 80% nucleotide identity, but other differences in the properties, once again in China. And then finally, what we see now that we have TGV circulating in pigs and PEDV, although these two are two different species of alpha coronavirus, we have seen now recombinants of this newly emerged PEDV and endemic strains of TGE, where they have a TGE backbone and they have the PED spike, and this allows them to escape immunity to TGE. So what we don't know is whether some of these SARS coronavirus 2 strains could recombine with any of the endemic particular beta coronavirus to create new virus strains. So this is a summary of some of the factors that can influence interspecies transmission of these enteric and respiratory coronavirus. One is the environmental exposure, the host reservoir interactions, where we have close contact with infected animals, infectious secretions or fomites. This can occur in wildlife interface with domestic animals, with pet animals, at the human interface, and also where there's human contact with bat reservoirs. This can occur in the live animal markets, as we saw for SARS and potentially SARS coronavirus 2. Up here is a picture of the raccoon dogs in the wild animal market in China, where there's multiple species, often in sanitary conditions. And it's interesting that it's not that PEDV came to the US from China, because the closest strains in the US were similar to Chinese strains, potentially in feed sources that were fed to pigs. So perhaps bone meal, or other feed sources. But this can also occur by environmental contamination, wastewater, sewage contamination. Another issue is the stability or dose of the agent in the infectious material and the transmission route. It's interesting that these beta coronavirus, bovine coronavirus, SARS, MERS, SARS coronavirus 2 are thought of as pneumoenteric viruses that can infect both the respiratory tract and gut. So this tells us that we can have possibly greater efficiency of transmission of combined aerosol and fecal oral transmission. And then the viral vectors, a major role there is the attachment entry with receptors, co-receptors. And like the original SARS coronavirus, the SARS coronavirus 2 binds to the ACE receptor shown schematically here on the surface of the cell. It actually has a higher binding affinity than for SARS coronavirus. And what's different completely from SARS was that this virus had a new furin cleavage site in the S1, S2, shown here, cleavage by furin, and that allows it to attach to the cell membrane. And even though this was unique to SARS coronavirus 2 and sarbecoviruses, but this furin cleavage site is common in many other viruses, coronavirus, including MERS, And also it was in a recently discovered Brazilian bat beta coronavirus. So this says this is not unique to SARS coronavirus 2 and could have been acquired in a recombination with the coronavirus. So another major factor in interspecies transmission, and I'll give examples with the alpha coronavirus, are recombinants, and especially common are recombinants in the spike. So we know that porcine, feline, and canine coronavirus, they're single species of alpha-1 coronavirus, and they're actually appear to be host range mutants of a common ancestral coronavirus, perhaps from wild feline or canine species. They have conserved APN receptors, so they can infect cats, dogs, and pigs that all have APN, aminopeptidase N. And so interspecies recombinants then can lead to new strains. And so this example is, first of all, the canine coronavirus type 2A. The dog virus is the ancestor for TGE. And in the spillover from dogs to pigs, there were mutations in the spike and also mutations in one of these accessory proteins, open reading frame 3B, and also a non-functional loss of 7B that's present in the original canine strain. And then we've also had this spillover of a second, of a TDE-like virus that spilled over into dogs, back into dogs. And this virus had part of the TG spike and the other genes that are similar to the canine coronavirus 2, but this was a new variant of canine coronavirus 2B. And then the most recent picture that we see here, it's this canine coronavirus 2B virus that is the backbone for the human pneumonia 2018 canine coronavirus that spilled over into humans. And this was discovered in 2017 to 2018 samples in hospitalized children in Malaysia and also from adults returning from Haiti. So these two strains, but interestingly, these strains had 99.4% nucleotide identity, even though they were from distant geographic locations. So these strains have the canine 2B backbone, shown here in the yellow, and they also have small deletion in the nucleocapsid protein, and a deletion in open reading frame 7B, likely rendering non-functional like in TGE. So what we have now is reservoir host species, dogs, cats, and pigs, a single alpha coronavirus 1 species that can maintain these strains and in which recombination can occur. And now we see there's at least occasional spillover from dogs to humans. So this is allowing the virus persist in a reservoir community and evolve within this community. So many of you probably heard about the feline coronavirus 23 strain in Cyprus. So this is another example of the alpha coronavirus 1 spike recombinant. There was a recombination of a canine feline alpha coronavirus 1, and it was occurred in Cyprus, and it's called feline coronavirus 23. It had altered tropism and virulence. It was a highly pathogenic spike recombinant with pantropic canine coronavirus, presumably from dogs, that spread to cats in Cyprus in 2023, had high transmission rates. It caused feline infectious peritonitis and deaths. In fact, it's estimated 8,000 cats died of FIP in Cyprus. So this recombinant virus had this feline coronavirus 1 backbone here, shown here, but it acquired the spike in a recombination event of a canine pantropic strain and a canine coronavirus 2 strain. And a very interesting aspect, like we saw with PRCV, and mutation from TG to PRCV, this large deletion in the spike near the N-terminal domain. And another interesting story is that these spike deletion strains and intact strains were co-circulating in cats. And some people think the intact strain was the one that circulated, but maybe these spike deletion mutant strains arose spontaneously in the cats and were what was associated with FIP. So we don't know the impact of this recombinant canine coronavirus 2 spike, the S1 N-terminal deletion, or the co-circulation of these strains with intact S or deleted S on FIPV transmission and pathogenesis. But this has been a highly unusual scenario because usually FIP strains do not transmit from cat to cat. Usually it's not, they arise spontaneously internally within cats, and then they're usually lethal in cats. So now I want to switch to the beta coronaviruses. We've worked on these for many years, and been using a germ-free calf model to study infections in calves and pathogenesis and immune response in vaccines. So a postdoc in my lab, Dr. Tsunemitsu from Japan, isolated these viruses, similar viruses to bovine coronavirus from wildlife, sandbar deer, white-tailed deer, waterbuck, and we sample from a child with diarrhea, and this sample, the HEC 4408, was very similar genetically to bovine coronavirus. So we wanted to test if these viruses could experimentally infect young calves, cause disease, and cause seroconversion in the animals. So all of the germ-free calves we exposed to these different viruses got diarrhea. They all shed virus and feces, and they all seroconverted with neutralizing antibodies to bovine coronavirus. So this was one of the first experiments to show that wild cervets could transmit coronavirus to cattle, and there could be spillover and spillback of an enteric strain in humans. And now we know that this is very important because on this basis, people started looking at white-tailed deer to see if they would be susceptible to SARS coronavirus 2 as well. And I'll talk about those studies. So now we have a beta coronavirus imbecovirus reservoir community with cattle that can transmit virus to turkeys, to avian species, two-way transmission with deer, with humans, and then related strains occur in pigs, horses, and a respiratory kennel cough strain in dogs. So I want to talk next about SARS coronavirus 2 in animals as potential new host reservoirs for secondary spillover to humans. So just to define these terms so it's clear what I'm talking about. So when these viruses spill over from an animal to humans, we refer to that as zoonosis. It's not clear if this could have spilled over directly from bats to humans. But what happened after this virus spilled over, it was highly efficient at human-to-human transmission, much more so than MERS or SARS. It's also postulated there could have been an intermediate host like there was for SARS, raccoon dogs or guns in the marketplace that could have transmitted the virus from bats to the intermediate host to humans. But then what we saw after this virus went on with all the different variants that arose, we saw spillback or reverse zoonosis and into multiple species. But I'm going to mention these four, and I'm going to talk mainly about mink and deer as examples here. But all four of these species, mink, deer, cats, and guinea pigs, were able to transmit the virus back into humans. And so now we have concern about animal reservoirs that could maintain SARS coronavirus 2. So we don't know if new or ancestral SARS coronavirus 2 strains will persist in animals with secondary spillover to other species. So you're all familiar with the scenario with SARS, where we had the original strains, and then there were key mutations, particularly this D614G that led the virus to be more transmissible or virulent. And then we had all these variants of concern designated by WHO, alpha, epsilon, beta, gamma, delta, and we're still having persistence of Omicron with over 500 sublineages. So all of these, and in the adaptation to humans and escape immunity, had a lot of key mutations, particularly in the receptor binding domain in the spike, as indicated here, and the most were in the Omicron. So was COVID-19 a zoonosis? There were many early cases that had contact with the Hunan seafood market in Wuhan, China. And it's now known that bats are likely the ancestral host reservoir for SARS coronavirus 2, and particularly these horseshoe bats or rhinolopus species. So multiple Asian bat coronavirus are related to human SARS coronavirus 2. Here's the phylogenetic tree. Two of these strains from China share high genome identity overall, but low identity in the spike receptor binding domain. However, after these strains were recognized during the course of the pandemic, strains were isolated from bats in Laos, and these strains are called Banal strains, and they had the highest nucleotide identity to the SARS receptor binding domain. They differed only by 1 or 2 amino acids, but more importantly, whereas these original bat strains could not bind to human ACE2, these strains could. And as I mentioned, we now know that some bat strains can also possess a receptor, purine cleavage site similar to the SARS. So these can also occur in bats in nature. So this suggests there could have been a role of bats in direct transmission of both SARS, where related strains exist, and SARS coronavirus 2. So we don't know still if there was an intermediate animal host that transmitted the virus to humans. Some suspect pangolins because, again, the spike receptor binding domain has a high nucleotide identity to SARS coronavirus 2 and can interact with the human ACE2 receptor. And then circumstantially, civets, raccoon dogs' DNA was found to be present in the Hunan seafood market, where SARS coronavirus 2 RNA was also identified. So the natural zoonotic versus lab source origin of SARS coronavirus 2 still remains controversial, but if you look at the scientific literature, a preponderance of peer-reviewed scientific publications have concluded that a zoonotic animal spillover is most plausible, and this was the conclusion of the most recent WHO SEGO report. So I'm sure many of you remember this stories in the newspaper and social media about SARS coronavirus spillover in animals when they were first detected. So some of the first detections were in mink in Europe, but they were also found in mink farms in the US. And then pets and households with SARS patients became infected through the human contacts, and particularly cats were more likely to be infected or show symptoms. And then we had all these outbreaks occurring in felids and other animals, non-human primates, in zoos. And then finally, we found it in free-ranging wildlife, white-tailed deer in Ohio, and that's what I'm going to talk about next. But then it even was found in rats in the sewers in New York. So they, again, potentially could play a role in transmission. So COVID-19 has been shown to result in two-way spillovers in mink. So there were a number of outbreaks in farm mink in Europe. shown that these outbreaks were introduced into the farm mink from humans, and then there was rapid transmission among these caged mink, so millions were culled or sacrificed. The mink showed respiratory signs. They shed virus, and some had interstitial pneumonia and died. So they had a syndrome very similar to the SARS coronavirus 2 syndrome in humans, like COVID-19. And then there was one case in Utah and 2 cases in Spain where it spread to wild mink, presumably from mink that escaped from the mink farms. And the next scenario, besides spillover from humans to mink, then it was found that mink could transmit the virus by way of reverse zoonosis back to humans. And this showed that the virus was transmitted to animal caretakers in the Netherlands and also Denmark. So in Denmark, there were 214 human SARS infections with mink-related variants. And there's one cluster that had 4 major genetic changes in the spike, with one associated with adaptation to mink ACE2 receptor. So this raised concerns for effects on transmissibility, diagnostics, and vaccines in humans, and this led to culling of additional mink. And then after this was recognized, and there was also found to be secondary transmission from the mink to other animals. So there were feral cats in the mink farms, and they were found to be infected, and some of these cats died. And then more recent studies have hypothesized that mink, possibly, especially wild mink, could transmit the virus to deer because a very ancient strain has a shared common ancestor between mink and deer was found in Canada. So there's also recent data from studies in Ohio from my colleague, Dr. Bowman, that Delta variant could transmit from farm mink to deer because mink-adapted mutations, this spike mutation in deer sequences within the mink delta clade. So this shows that it may be necessary to have animal vaccines to prevent new variants and spillovers, protection of mink, hamsters, deer, cats. And there was a spike adjuvanted vaccine developed for mink and zoo animals, but there's very little efficacy testing of this vaccine. So going on to deer as a possible reservoir host, in the United States, it's estimated there are 30 million deer, or 10 deer, 1 deer for every 10 people in the US. So a very dense population of white-tailed deer. They live on our farm. They live in the woods, so we see them almost every day. The deer were experimentally infected in 3 different studies. There was virus transmission to contacts, but no disease and seroconversion. So subclinical infections, as you would expect in a reservoir host. The deer were naturally infected with SARS coronavirus 2. It was later proven. First, it was shown there were antibodies in 40% of deer surveyed in the East and Midwest. But then our studies in Ohio that we conducted in 2021, that's when this B1.2 virus was most prevalent. And we sampled deer during this time just before the alpha strain was starting to take over. And we found that 36% of the white-tailed deer in Ohio, and Dr. Pucciapucci showed 33% in Iowa were shedding SARS coronavirus 2. So what we found was there were at least 3 introductions here from humans to deer based on the different strains. There was evidence of deer-to-deer transmission for at least 3 of these outbreaks that we tested. And then after this, multiple studies showed that all of these different variants could be transmitted from humans to deer, alpha, gamma, and Omicron. And then there's at least 2 studies that suggest there was deer to human spillback in one study of this novel strain in Canada, but also a Delta coronavirus in the US. So we know now that alpha, delta, and gamma and deer variants can co-circulate even after these cases are gone in humans. So we were out here surveying when we found these delta. The alpha strains had disappeared in humans, but we were still picking up later when we were surveying after 2021 these alpha strains in deer. So this could mean that the deer could be a reservoir for strains that persist in deer but are no longer in humans, or new strains could arise in deer that could possibly be transmitted to humans. So this is why we need a One Health approach to monitor cervids, other animals in the global ecology. We don't know how deer acquired the SARS coronavirus 2 from humans. We don't know if the virus will persist and adapt in deer, and if deer will transmit the virus back to humans or other wildlife, such as mink or any of the other susceptible ones, or to grazing ungulates, cattle, sheep, or goats. So we now know that the SARS coronavirus can naturally infect at least 60 species of animals, all kinds of wildlife, zoo animals, domestic animals, pets, and also there's high susceptibility in at least 24 species that have been confirmed. What we have seen then is that there's deer-to-deer transmission in white-tailed deer, and there's two ways, spillover and spillback, from humans, spillover to the deer and spill back into humans. Also the same for cats, for the farm mink, and in a study in Hong Kong, also for pet hamsters. So the lessons learned for One Health is that SARS coronavirus 2 has a broad host range. It can infect pets, zoo animals, mustelids, wildlife. It's mostly subclinical infections, except for farm mink and cats. So this raises concern for animal health. The spillover and spillback from cats, white-tailed deer, and farm mink and hamsters, but the pathways remain unclear. And again, we don't know if maybe eventually we need animal vaccines to prevent new variants and spillovers. So the concern is, will the virus persist, adapt, and mutate in wildlife that transmit new variants back to humans, livestock, or other animals? And this would then establish a new host reservoir with all these species and SARS coronavirus 2. So WHO has predicted the next pandemic could be disease X. And from all we know now about coronavirus, it may not be what if, but when there may be future coronavirus transmissions to humans. So the red flags here are the SARS-related beta coronavirus in bats and the MERS that's still circulating in camels. We've had 3 prior pandemic epidemic events with SARS, MERS, SARS coronavirus 2, but some of these related strains still persist in bat reservoirs, and we know that there's ongoing transmission between camels and humans in the Middle East for MERS. Then we have the porcine Delta coronavirus scenario. This was discovered in pigs in Hong Kong in 2012. It got to the US in 2014, caused an epidemic and killed a number of baby pigs. It has a broad host range. It can infect avian species, swine and ruminants. It can infect human cells. It can use the aminopeptidase N receptor, like for the alpha coronavirus in swine. And there were infections in 3 Haitian children with mild febrile illness with 2 different lineages, most similar to porcine Delta coronavirus. So now occasional spillover to humans. And then we've recently started to conduct some serologic studies. Dr. Kinney's lab in our Center for Food Animal Health. And they collected samples from youth in close contact with swine. These actually samples were collected by Andy Bowman to look at swine influenza. So these were tested in ELISA and virus neutralization tests, and 9 out of 100 came positive for antibodies to porcine Delta coronavirus, suggesting this virus can and does circulate in humans. I already mentioned the scenario with canine alpha coronavirus spillover to humans 2017-2018, which is another potential WHO disease X. So finally, I just want to mention the One Health targeted surveillance at the human-animal environmental interface so that we would have opportunities for better surveillance and containment. The slide is adapted from Dr. Koresh, where we have spillover from wild animals, animal vectors, potential intermediate hosts and domestic animals, amplification, and then possible spillover into humans with human-to-human transmission and amplification. So what we need to be aware of is try to detect the possibility of a human epidemic early before it can spread worldwide, as the case with SARS and SARS coronavirus 2. We also need to have methods for early detection of spillover events. So this would be at the animal-human interface, particularly at wet markets, but even with domestic livestock, pet animals. And then we need generic measures to reduce the chance of zoonotic spillover source control, where we have deforestation and any situations where it exposes wildlife human interface with possibility of disease transmission. So this is essential for situation awareness, risk assessment, understanding pathways of spillover to allow interventions that can reduce risk. So again, how will we mitigate coronavirus pandemics? We need a One Health focus. We need to prioritize the target animals. We need environmental surveillance, even on farms with manure pits, wastewater, sewage. We need to look at environments with contacts with infected animals and secretions or fomites, wildlife, domestic animal, human interface, bat caves, bat reservoirs, live animal markets, fecally contaminated food sources, environmental contamination. We need human surveillance for infections by animal coronavirus, respiratory, enteric, and outpatient clinic, symptomatic hospitalized patients, like was done for canine coronavirus detection, immunocompromised patients. We need to develop animal vaccines as needed to prevent new animal host reservoirs. And most importantly, we need funding mechanisms that can support integrated collaborative research on zoonotic diseases that will include humans, animal reservoirs, and the environment. Some of these are proposed in these different references here for how to handle this. So we need a One Health focus to control zoonotic diseases with the combined efforts of veterinary, medical, environmental, cross-disciplinary scientists, and global interagency collaboration. So just to summarize, the coronavirus continue to evolve and emerge globally as a cause of fatal respiratory and enteric infections of animals and humans. They're genetically diverse with frequent mutations, deletions, recombination, but the genetic basis for interspecies transmission, changes in tissue tropism and virulence are uncertain and less well understood. The epidemiology and spillback of SARS coronavirus 2 from humans to animals or reverse zoonosis, it's unclear how this occurs, as is the potential for secondary spillover to humans from new SARS coronavirus animal hosts, such as the white-tailed deer, the mink, and some coronavirus, as I mentioned here, the canine coronavirus, human canine coronavirus, and human porcine delta coronavirus may be poised for zoonotic disease outbreaks and pandemics, WHO disease X, if they're able to acquire sustained human-to-human transmission. So all of this indicates that we need One Health approaches to study zoonotic coronavirus. With that, I'd like to thank my lab and collaborators, especially Dr. Vasilova, who worked with me on who actually discovered the human canine coronavirus with Dr. Gray in Texas and did the sequencing on the virus and continues to study it. Previous postdocs and students in the lab, Dr. Chong Wong, who we work with porcine coronavirus, and then Dr. Scott Kinney, who've been working with porcine Delta coronavirus. And with that, I'll be happy to answer any questions.
Thank you very much, Linda. Very clear.
Thank you.
Abrimos entonces un espacio de preguntas. La doctora Linda Saif nos invita a que las hagamos. Les voy a pedir, si son tan amables, o que levanten la mano o que me escriban en el chat. Pido palabra Pepe, entonces voy anotando el orden de las preguntas. Alejandro, tal vez vos quieras romper el hielo.
Bueno, thank you very much, Linda, for your presentation. Excellent, excellent. Two main things I will acknowledge from your presentation. The first one is the approach of One Health to study and understand this important complex situation. It's not One Health is not in the world, but this is a typical example for an infectious disease. So thank you for giving information, basic, validated information to approach this as a One Health program. The second question is a general one, but is strongly related. Why coronavirus have this great host range of susceptible species? Do you know any other family of viruses that has this widespread susceptibility in the animal and human population?
Yeah, I think that's a really good question. And we don't really know the answer to that. But these beta coronavirus that I mentioned, particularly the SARS, the bovine coronavirus, and the SARS coronavirus 2, and even MERS, all of these, for some reason, have very broad host ranges. So one possibility is that some of these viruses, besides a protein receptor like the ACE2,
APC, yeah.
or aminopeptidase N and things like that, the bovine imbecovirus also have sialic acid receptors. So this is possible that this will expand the host range of these viruses. But we really don't understand why SARS coronavirus 2 has this amazingly broad host range, other than some of these, the ACE2 receptor is shared broadly where the virus binds across the number of animal species. And this was shown with in silico modeling for the receptor and interaction with SARS coronavirus 2 spike.
And another question is related with the bats and the role of bats in this particular infection. Have you been able, or any laboratory in the world, has been able to trace back for how long this coronavirus has been detected in bats, in the bat population?
So there are a number of people who study these viruses in bats intensively in the bat caves and so forth. And I think that for the most part, many think that these viruses persist in bats, or they have essentially repeated infections in bats, so that the bats, like when they have the young, the offspring, more likely to shed at certain periods of time. Maybe they're more likely to shed shortly after hibernation or during hibernation. All of the logistics of that are not well recognized. But when they sample these bats, they do find that these coronavirus are persisting in these bats.
Without causing disease.
No disease.
No disease.
This is the usual scenario for a reservoir host. The same way, there's a huge variety of delta coronavirus in the avian species, particularly songbirds and shorebirds. And so far, no one has shown disease with these delta coronavirus in the avian hosts. Very similar to some of the influenza strains.
Influenza, yeah.
Yeah. So they're carriers. The bats appear to be carriers, but not clinically infected. And the birds appear to be carriers, and probably some rodents too are carriers for some strains that maybe do not cause disease in the rodents.
Okay, thanks, Linda.
Linda,
Yes.
there is a question in the chat. It's in Spanish, so I will try to translate it to you. It's from Dadin Moore. Says your presentation was excellent, and thanks you a lot, and has one personal question to you. First of all, how do you protect yourself and your family from any infection from coronaviruses, and from your family and your pets, since you are all day working with them in your lab?
So during COVID-19, I wore a mask, and everyone in the family wore a mask. So we were very concerned about PPE and rigorous and stringent hygiene measures and things. But likely, from having worked with all these coronavirus over the years, and my husband also worked with infectious bronchitis virus, I think we've all acquired some degree of immunity. But I mean, that's a good question, and it's just, a public health question in terms of distancing, wearing masks, and hygiene. Was about all we could do till the vaccines came in. And then once the vaccines came, there was still transmission, but at least you were less likely to be hospitalized.
Okay, so the second question that he made was related to the vaccine strategy. He says also as
would be necessary. I can do it, Emilio. Would be necessary to design new vaccines according to the host?
So it appears that the spike vaccines are working very well, whether they're subunit spike vaccines or DNA or RNA spike gene vaccines, receptor binding domain vaccines. The RNA vaccines are too expensive right now to really be able to use in animals unless they're an extremely valuable animal. But certainly, Zoetis came up with this subunit vaccine that seems to have worked in the zoo animals. I don't know much about the feedback in the mink, how well it's worked in mink. In theory, if you identify the spike and the receptor binding domain, and you're able to make a RNA or a subunit vaccine, and then with the subunit, you usually have to have an adjuvant, an effective adjuvant. You should be able to also get protection in animals as well as humans. I mean, the big vaccine concern and big question now is blocking transmission. And so everybody's going back and trying to design intranasal vaccines. But there's not enough support right now for these vaccines, unfortunately. So, and I mean, one needs side-by-side comparison between those vaccines and the intramuscular, but there are intranasal vaccines for influenza that are thought to work well, at least in young people. But that's the direction this is going in terms of future vaccines for SARS coronavirus 2 or even other coronaviruses that have a respiratory pathogenesis.
Ok, pienso que eso ha contestado las preguntas de Darin Moore y Irene Álvarez. Si quedó alguna duda, pueden decirlo en voz alta.
¿Puedo?
Sí, claro, Alejandro.
Linda, it came up to my mind after seeing your presentation a very pertinent question. Did the regulatory agencies on the animal area have taken any measure in the United States or elsewhere with regard to the potential cross of the virus in susceptible species? I am speaking in relationship, for example, in the intensive production area on birds or pigs, in which in some cases they use the bed of the husbandry of the birds, for example, to make food for other species, because like pigs, because the high protein content of the bed of birds, for example. And this could be a potential risk of transmission in between species.
Yeah. So after they did some work and thought that there was the chance that porcine epidemic diarrhea might have come in on swine feed, some kind of swine feed product from China, then they started paying more attention to this component. So how it was processed, and then testing to see if they found virus in it, infectious virus or anything. So I think there's that awareness now that they're paying more attention to possible feed contamination. But, we don't even know the tip of the iceberg here, because, China is second or third in fur farms. And, they likely feed offal, the guts of animals to the mink. And we don't even know what was happening in these farms in China. And we may never find out either. But, that's another way that the virus could spread. Like you said, if you're feeding dead animals to like mink or something, then obviously if those animals you're feeding them are infected, then obviously that's a good chain of transmission for the mink.
Yeah, sure. Thanks, Linda. Sí.
Yeah.
Gracias.
Doctor Saif, voy a hacer una pregunta, Alejandro. In agronomy, we use the bed from raised intensive cattle production or bird we use them as fertilizers or to provide nutrients to the soils. We disperse them in the soil. We disperse them in the soil as a fertilizer source. Do you think that could be also a way to transmit diseases, virus diseases, to other species that fit from?
I think that's something that really needs to be examined. I mean, most of the time, the idea here is to have the manure pits or compost, and dead animals are usually composted, in which case the heat will destroy a lot of these membrane-bound viruses. But if you had a very stable virus that's, more resistant, then that's a possibility. And I think, just have to look into those soil samples and see if any virus can be recovered or persist. in the
Linda, I am thinking, I'm sorry, Emilio, and audience, I am thinking in not Deutschland, Holland, the north of France, and the east of Germany, in which the practice of distributing directly the fluids from the high-intensive production of porks is, dispensed on the as a fertilizer. So if you have an infection there, you have a spreading this coronavirus for everywhere. You see.
I think most people think in the manure pits, if it's done
Manure, that's it. Yeah.
Yeah, in the manure pits, if it's done properly, like compost,
the
heat will be enough to inactivate these more fragile viruses. But the question is, will it really inactivate all more stable viruses? And that's probably what should be, tested more. I mean, so, one of the big ways to monitor the spread of SARS coronavirus 2 became wastewater surveillance, because the virus, the RNA was detectable in wastewater. So you could see when the wastewater started to peak, you knew that you were likely to get the spread of this next variant, and we actually used that at the university in the dorms to look, what was occurring and what variants were there, and when you might need more public health measures to be implemented, or you might need to temporarily close the university and that thing when you see these peak. So, we definitely could do more wastewater and near pit monitoring at animal farms. We have the technology now to do that. And even animal farms, you can put detectors for aerosols and monitor what you're getting from the aerosols in these filters. So, all of these things really can be implemented now, and probably should be.
Okay. Thank you very much, Linda. Linda, there is a question from Santiago Raggio in the chat. It's written in English. And would you like to read it, Santiago? ¿Quieres leerla?
Okay. Good afternoon. Thank you very much, Linda, for the presentation. I have a question related to vaccination in dogs. Do you recommend vaccinate dogs with inactivated vaccines like we have available nowadays?
So are you talking about the SARS coronavirus 2 scenario, or you're talking about general scenario?
No, I'm talking a general scenario with the canine coronavirus vaccine.
Oh, canine coronavirus.
Yes.
Yeah. So if you have that pantropic canine coronavirus, it's systemic, and it's going to the lymph nodes, and it's probably associated with viremia, then potentially intramuscular can have an impact there if you're using proper adjuvants. But if it's the enteric coronavirus, our studies in swine suggest that you really need oral vaccines to be highly effective against enteric infections as opposed to systemic. The one scenario that would change things, though, that we also found, if animals were naturally infected and orally primed, we could get a very good even mucosal response with even just an intramuscular booster, because the animal had already been primed mucosally. And presumably those cells then reside in other tissues besides mucosal tissues, in some of the lymph nodes and things that you can re-stimulate and still get a good booster response. So for a primary immune response, I'm of the opinion that we really, to prevent enteric infections, we need oral vaccines, and for respiratory infections, intranasal vaccines may be more effective because of the mucosal immune response.
Thank you very much. Is there any other question? No, creo. I think there are no more questions, Linda. The time is over and we are on time. So at last we have only to say thank you very much for coming to this space. Thank you very much for your very clear presentation, to be with us and to be so kind answering all our questions. Alejandro, would you like to say anything else to
Oh,
Linda?
thanks, Linda.
Oh, you're quite welcome. It's
Linda.
great to see all my colleagues. Fernando, who worked with me in the lab, and Ana Sadir, who worked with me. And it's great to see all you guys again. And just stay healthy and stay in touch, I hope. So thank you for inviting me.
Okay.
Thank you very much, Linda. Thank you very much. And
Bye-bye.
Gracias a todos por participar y por estar. Nos vemos en la próxima conferencia. Ya saldremos.
Muy bien.
Hasta luego. Muchas gracias. Bye bye, Linda.
Thank you.
Thank you very much indeed.
De nada.